detects whether anti-nuclear antibody is present in the patient blood sample or not. In order to do so, the technicians withdraw blood from the patient, and it's basically a simple blood test, requires less than 10 ml blood to be drawn from you. So you don't need to even fast for the ANA test. But what you need to know is what to expect from an ANA test result. In most cases, positive ANA test means that you might have an autoimmune disorder. So it's a screening test. But many people do have ANA test positive but are very healthy. So how to interpret the result? And in this video, we'll talk these things in detail. Particularly women who are older than 65 shows elevated autoantibodies and their test is positive for ANA. But what does all these means in terms of autoimmune disorders? Let's say this is a patient who is suffering from systemic lupus erythematosus, and based on this particular characteristic rashes in her face, you can understand it might be a symptom of lupus. So in order to get 100% sure, the doctor prescribes an ANA test as a beginning. So in that particular test, blood samples would be drawn from the patient, the serum would be extracted, and the serum would be added to a Hep2 cell line in a pathological laboratory. Now, Hep2 cell line is an array of hundreds of antigens. Now, if the anti-nuclear antibody is present, most likely these antibodies would detect antigens present on these Hep2 cell lines. Hep2 cells are used because they have a wide variety of these antigens, and these varieties are not recapitulated in many other cell lines. But luckily, Hep2 cell line does. So we have two outcomes. One outcome is a patient sample has anti-nuclear antibodies, and second outcome is the patient sample might not have any anti-nuclear antibody. So obviously, the second case, the ANA test would be negative, and in the first case, the ANA test would be positive. So what we do is, after incubating the Hep2 cell line with patient-derived serum, we add secondary antibodies. So let's say particular anti-nuclear antibody was present, and against that, a secondary antibody is used, which is fluorophore tagged. So if the primary antibody was present in the patient sample, only then the secondary antibody would bind to the primary antibody and it would show the fluorescence. Otherwise, it would be washed away in the washing stick. Ultimately, scientists actually look at these particular data under fluorescence microscope. So by looking at the cells under fluorescence microscope, pathologists can understand what is the scenario. There are many staining patterns that can be obtained, such as homogeneous, where the overall fluorescence is coming from all the parts of the cell. There could be speckled pattern where the fluorescence is like highly dot-like structures. There could be peripheral ring-like structures and nucleolar structures present from the, all these are possibilities that one can see from an ANA test. There could be no fluorescence at all. That's the negative test. Now let's see what does this ANA pattern means. In all these circumstances, we can understand the ANA test is positive. So looking at these test results and looking at the clinical databases, pathologists can comment on what possible disease might be there. But it's not a sure shot confirmatory test. It's basically a screening and an assumption. Further tests are required. For example, if you have a homogeneous ANA pattern, then it might be SLE, might be DLE, might be rheumatoid arthritis as well. But if the pattern is speckled, most likely it could be also MCTD, scleroderma, or even systemic lupus. So further tests are required. So all these patterns help us to get some lead or some idea that what further tests should be done to get a confirmatory result. So what we understood from these discussion that ANA is basically a screening test. It's not a confirmatory test for any disease. It only tells us that you might have autoimmune disease or might not. Because we also saw that ANA positive test doesn't mean that you are having an autoimmune disorder. While ANA test can't confirm specific antibodies, it can rule out some other disease that you are not affected by this and that. So it's kind of a good screening method. Further tests need to be always done along with ANA to predict one particular type of disease.